everyone, my name is Alaisam Sulaiman, and today I'm going to discuss about what is gametes, what is meiosis, the formation of male's reproductive cells, the process of spermatogenesis, and the purpose of spermatogenesis. So without further ado, let's get started. So first, what is gametes? Gametes are an organism's reproductive cells. They are also referred to as sex cells. Female gametes are called ova or egg cells, and male gametes are called sperm. Gametes are haploid cells, and each cell carries only one copy of each chromosome. These reproductive cells are produced through a type of cell division called meiosis. Next, what is meiosis? Meiosis is a type of cell division that reduces the number of chromosomes in the parent cell by half and produces 4 gamete cells. This process is required to produce egg and sperm cells for sexual reproduction. During reproduction, when the sperm and egg unite to form a single cell, the number of chromosomes is restored in the offspring. Formation of male's reproductive cells. Spermatogenesis is a process of developing male gametes, known as sperm within the male reproductive organs, the testes. In this process, each sperm, haploid containing a single copy of each chromosome, in order to create the haploid gamete, a cell undergoes the process of meiosis in which the genome is replicated and divided twice to produce 4 haploid gametes. This process generally occurs in the seminiferous tubules of the testes following different stages. It is followed by the maturation in the epididymis, where they are secreted in the form of semen along with glandular secretions. This process begins during puberty and ends only when the individual dies. The complete process of spermatogenesis in males are carried out by the actions of Leydig cells, hypothalamus, and pituitary gland. The quantity of these sperms gradually reduces with the age and finally leads to infertility. Stage 1. The diploid spermatogonia is situated in the seminiferous tubules, which include twice the total number of chromosomes. This replicates methodically in interphase before the method of meiosis 1 to create 46 pairs of sister chromatids. Stage 2. In this, the chromatids allow the exchange of genetic information through the synapsis process. It is done before dividing into haploid spermatocytes through meiosis. Stage 3. In this division, the new 2 daughter cells will further divide into 4 spermatids having unique chromosomes that are approximately half in number to the original spermatogonium. In stage 4, the cells move from the lumen of the testes to the epididymis. They get mature and developed into 4 sperm cells with the growth of microtubules on the centrioles to develop an axoneme. The remaining centrioles elongate and develop into sperm tail. So now, what is the purpose of spermatogenesis? The process of spermatogenesis occurs to create mature male gametes, which then fertilize female gametes to create a zygote, a single-celled organism. This results in cell division and multiplication to create a fetus. For a healthy offspring, the number of chromosomes must be maintained properly across the body, as failure can lead to some abnormalities.
Formation of female's reproductive cells. Oogenesis in the human female reproductive system is a growth process in which the primary egg cell or ovum becomes a mature ovum. In any one human generation, the egg's development starts before the female that carries it is even born 8 to 20 weeks after the fetus has started to grow. Cells that are to become mature ova have been multiplying, and by the time that the female is born, all the egg cells that the ovaries will release during the active reproductive years of the female are already present in the ovaries. These cells, known as the primary ova, number around 400,000. The primary ova remain dormant until just prior to ovulation, when an egg is released from the ovary. Some egg cells may not mature for 40 years, and others degenerate and never mature. Oogenesis occurs in the outermost layers of the ovaries. Oogenesis starts with a germ cell called ooganium and undergoes mitosis to increase in number. The process of oogenesis takes place in the following 3 stages: prenatal, antral, and pre-ovulatory. Prenatal stage. The primary oocyte grows while being arrested in meiosis 1. The follicular cells proliferate and form a stratified cuboidal epithelium. Such cells are known as granulosa cells. These cells secrete glycoproteins to form zona pellucida around the primary oocyte. Antral stage. The fluid-filled area present between granulosa cells combines to form a central fluid-filled space called the antrum. These are known as secondary follicles. In every one cycle, these secondary follicles develop under the influence of follicle-stimulating hormone and luteinizing hormone. Pre-ovulatory stage. This stage is induced by LH surge, and meiosis 1 completes here. 2 haploid cells of unequal sizes are formed within the follicle. One of the daughter cells that receive less cytoplasm forms a polar body. This cell does not participate in ovum formation. The other daughter cell is known as the secondary oocyte. The 2 daughter cells undergo meiosis 2. The polar body replicates to form 2 polar bodies, while the secondary oocyte arrests in the metaphase stage of meiosis 2. Purpose of oogenesis. Oogenesis helps to retain sufficient amount of cytoplasm in the ovule, which is essential for the development of early embryo. Formation of polar bodies maintains half number of chromosomes in the ovule. What is fertilization? Fertilization is the process of combining the male gamete or sperm with the female gamete or ovum. The product of fertilization is a cell called a zygote. Fertilization occurs when male and female